Good morning. This is part 6 of heredity, and I hope this will be the last recorded session. Whatever slides remain will be discussed in the classroom next Thursday, inshallah. I will be starting by this slide because I discovered a small mistake at the end of the last video when I said that this ratio, which is 20 over 64, which represents that point of the curve, okay? I said that this represents the darkest of the skin colors. Actually, it represents the mulatto color, because here, what is written here is the number of dominant alleles. So here we have A capital, A small, B capital, B small, C capital, C small. So in every pair of alleles, we have only one dominant allele, or one of them only is dominant. This means that half of the alleles are dominant and half of them are recessive. So if the alleles for dominant represents the highest level of pigment formation and precipitation, then if you have only 50% of your alleles dominant, then you have an intermediate skin color. But here, the number that is written here, which is 6, indicates that all of the 6 alleles in the 3 pairs are dominant, and this represents the black color. So when you have all of your alleles dominant, then this means that you are the darkest, or you have the darkest skin, and your skin color is black. When you have only 3 dominant alleles, this means that you have a mulatto skin color. And if you have 0 dominant alleles, this means that all your alleles are recessive. The 6 alleles on the 3 pairs are recessive, and this indicates that you are white-skinned. As in any curve, the majority is the intermediate form of the trait. So here, the majority of the individuals, which are 20 over 64, have the intermediate skin color, whereas the extremes, having 0 dominant alleles and 6 dominant alleles, represent only a minority. Since you see here, their number is very close to 1 amongst the 64. So you have one individual out of the 64 that is, or one probability out of the 64, that the probability is 1 over 64 for the individual to be white, and for an individual to be completely black, having the 6 alleles dominant, is also 1 over 64. And this curve applies to any trait where you have a gradation, or a gradual increase in the intensity of the trait or the phenotype with the number of dominant alleles. So it applies to skin color, it applies to hair color, and it applies to the height of the individual. The more you have of the dominant allele, the more pronounced and intense the trait is, because the more you have of a dominant allele like A, the more you produce of the protein, and that protein is a pigment, and that pigment is a protein molecule that gives color, or it might be a hormone, for example, that is concerned with the height of the individual. We call this curve a bell-shaped curve. Now we're going to talk about a very rare condition in genetics and medicine that is called genetic Mosaicism and genetic chimerism, and the difference between the two. As you know from the structure of the cell membrane, mosaic means that it has patches of phosphate heads and patches of lipid tails, areas that are hydrophilic, areas that are hydrophobic, in addition to the presence of the embedded protein molecule. So anything that is patchy, that is heterogeneous can be called mosaic or showing Mosaicism. Whether we are talking about Mosaicism or Chimerism, we are talking about a single organism, but within that single organism, we have cells that have more than one distinct genotype. Meaning that if I say that an individual, a pea plant, for example, has the genotype R capital, R small for the flower color, then all the somatic cells of that pea plant will have this genotype for that characteristic, which is the flower color. But if you find that in some patches we have, or some areas of the stem of the plant, for example, we have the genotype R capital, R small, and in others we have the genotype R small, R small. This denotes a condition of Mosaicism, where a single organism has different genotypes in different parts of the organism. Of course, I'm just giving you a theoretical example, meaning that the stem does not contain a color of the flower, but whatever the characteristic is, then you have patches where you have R capital, R small, patches where you have R small, and other patches where you have, for example, R capital, R capital. This is genetic mosaicism. It is the presence of two or more cell lineages. A lineage is a cell type that is committed to a particular genotype. So if you have different cell lineages, then you have different genotypes, but the organism still arises from a single zygote. But if this patchy genotype, or this presence of different types of genotypes in the same individual, results from two distinct cell lines or two different organisms, then it is called chimerism. So in Mosaicism, it's a single zygote that developed into a whole organism, but inside the same organism, there are patches of cells with different genotypes. But in chimerism, we have two distinct cell lines derived from two different organisms. So this might result from the merging, for example, of two zygotes. So we had two different zygotes with two different genotypes, and then those two different zygotes with two different genotypes merged together and formed one big zygote. So both genotypes will be represented in the zygote. This also can be seen whenever we have organ transplantation, because if you give, for example, a certain organism, if you have a liver that is diseased and you transplant a part of another liver from another organism, then this would result in a case of chimerism. In such a case, the origin is different. It's not a single zygote where you have two cell lineages, but they are two zygotes that fuse together, and therefore every zygote is coming with its own lineage, or it's two organs that are being fused together because of organ transplantation. For example, when we transplant bone marrow is a tissue inside the bone cavity, the bone marrow cavity, that is responsible for formation of blood cells. So if you transplant bone marrow from a donor, and that donor is, of course, a different individual, then all the blood cells that will be produced will give rise to different blood types. Sometimes the chimerism is shown as patchy areas of different phenotypes, meaning that the genotype will be expressed on the phenotype. Of course, here, concerning the blood cells, when I say that the blood cells are of different blood types, there could not be major differences when you implant or transplant bone marrow. For example, if the individual is an AB individual, or let's even say that because AB is a universal recipient, let's talk about an individual whose blood group is O. Blood group O doesn't have any antigen on the red cell membrane, and it can receive only of blood group O. So where are the differences that we are talking about in the phenotype? This might be due to differences in the MN group, for example. As you know, one of them might be MM, the other might be MN, for example. And in such a case, these light variations between the genotypes of the individuals would not react in would not result in incompatible blood transfusions, because, as you know, at the body temperature, there are no interactions between the MN antigens and the antibodies that are present in the plasma. But of course, we cannot have major variations. For example, we cannot give blood cells or bone marrow that produces blood cells of the AB type to an individual that is of group O. Sometimes, as we said, chimerism involves the fusion of zygotes. So if the zygotes are of different sexes, then we would have both male and female sex organs, and this is one example of hermaphrodite or intersexuality, where the organism might have both the male and the female sex organs. And now we're going to talk about the genes and the environments. As we said before, there are two factors that affect a trait. The first factor is the gene, and the second factor is the environment. So in order for a gene to be expressed, you should have the suitable environmental conditions for that gene to be expressed, meaning that the environment is very important in determining the way the gene will be expressed. There is a term that's not written on the slide, but that I will be writing now, which is plasticity. Plasticity. And plasticity simply means that you have a phenotypic change, meaning a change in the trait or a change in the phenotype, and that phenotypic change results from environmental factors. So it is a phenotypic change that results from changes in the environment. And on this slide, we are going to see different examples for environmental changes that might result in phenotypic changes. And furthermore, we are going to study certain human diseases that are due to not only a genetic component, but also environmental influences. For example, if you talk about heart diseases, you might be genetically predisposed to a heart disease, having the genes that allow you or that might result in coronary artery disease, but you are on a healthy diet and you exercise regularly, so you don't get the disease. Diabetes is another disease that runs in families, meaning that for you to be diabetic, especially in type 2 or adult onset diabetes, you should have some kind of genetic predisposition to the disease, but it all depends upon your lifestyle, upon the stresses you are exposed to, the type of the diet you eat, the lifestyle you lead. Are you exercising regularly or not? And the same applies to all the rest of the disease. For example, in cancer, it is related to pollution of the environment. In schizophrenia, bipolar disorders, and all other major psychological diseases, the person might be genetically predisposed, but in order for the disease to show symptoms, the patient would be exposed to stress. For example, in females, pregnancy is a stress, breastfeeding is a stress, exposure to childhood trauma, for example, is a stress, and so on. Okay, so the environment can alter gene expression, as we said, and this is what we call plasticity. For example, in fruit flies, we have two types of wings, as you know, the long wings and the vestigial wings. And as we will see in several genetic problems, the vestigial wings are recessive, and the long wings are, or the presence of the long wings is controlled by a dominant allele. So the dominant allele controls or is expressed as long wings, whereas the recessive allele is expressed as vestigial wings. But it has been found that certain recessive organisms that are homozygous recessive, for example, having L small genotype, and if the eggs of the Drosophila fruit fly are incubated, for example, at 25 degrees Celsius, they might grow long wings. How does this occur? How does the environment affect wing length and other characteristics? And how does temperature in particular affect the expression of the gene? As you know, the gene is a DNA segment, and that DNA should be first transcribed into, so we have the process of transcription in order to get messenger RNA, and then messenger RNA would be translated into a protein. So here you have the process of translation. And whether you're talking about transcription or translation, you need enzymes for both. So the temperature actually affects the formation of a protein by affecting the enzymes that control transcription of DNA into a messenger RNA molecule, and then translation of the messenger RNA molecule into a protein, and that protein will be an enzyme or a hormone that regulates the characteristic. The same applies, for example, to temperature, to intelligence in human beings. Maybe you have genes for being intelligent, but you are not raised in an environment that helps you develop this intelligence, whatever kind of intelligence it is. And this always raises the issue of nurture versus nature. Nature is the genes you have, and nurture is the nutrition and the way you are raised and all the environmental factors that affect the expression of the genes. So in order for DNA to give a protein, this is what we call expression of the gene or gene expression. This is what we always call from gene to protein. From gene to protein, the process is expression, is gene expression. Here we have three types of fruit flies. We have the fruit flies with the normal wings in letter A, normal fruit flies, and we call them the wild types. So when we say the wild type fruit fly, then we mean the ones that have the regular wings. And then here we have the homozygous recessive ones. And here, this is the vestigial wings result from a recessive mutation. So if both if you are homozygous recessive, meaning that you inherited two mutant recessive, not you, but the Drosophila fruit fly inherited two recessive alleles, then it would have vestigial wings. There is another abnormality of the wings that is called curled wings, and actually they're not curled in shape, but they are curled in their direction. As you see in letter C, they don't have the normal direction towards the posterior part of the insect, and this curly winged fly should have received a dominant mutation. So contrarily to letter B, where we have the vestigial wings because of a recessive mutation, we have the curled wings due to a dominant mutant gene. And since it is a dominant mutant gene, then it is enough for the fly to receive only one dominant mutant allele in order to show the characteristic, whereas for the recessive mutant allele of vestigial wings, the fly is supposed to receive two. It should be homozygous, but in curled wings, it is enough for it to be heterozygous. Now we're going to talk about something very important that we already have been discussing for a while, which is epigenetics. Actually, when the temperature affects the expression of a gene, you have a different phenotype. But that different phenotype that you obtained, or the change that occurred in the phenotype, is not due to a change in the genome itself. It's neither due to a change in the number of chromosomes, nor due to a change in, for example, the sequence of nitrogenous bases in a gene, but it is due to a factor that is not related to the genome, to the original genome structure, the nucleotide sequence, and chromosomal number. Like, for example, temperature. When the expression of a gene changes because of a change in temperature, you haven't made any change in the genome, but all what you did is that you changed how the genome was expressed into a trait. And this is what we call epigenetics. So epigenetics includes a change in phenotype, a change in gene expression, because in order for you to obtain a phenotype, the gene should be expressed into a protein. And this alteration in phenotype occurs without any alteration in the genome. And of course, if there is an alteration in the phenotype and an unexpected or an unusual actually alteration in the phenotype, and you have a certain genome, you are expecting a certain phenotype and you get a different phenotype because of the environmental changes, and therefore you get non-Mendelian results or results that do not fit with Mendel's ratio. We have already discussed one, which is the effect of temperature in determination of wing length in Drosophila. We also have another example at the end of the presentation that we will not be discussing now because it's a problem that it's a problem on AP Classroom that you will be solving as homework. And then we also have the effect of bisphenols, for example. As you know, bisphenols are mimics of estrogen on a male fetus. We have said that if a male is a normal male having the chromosomal structure XY, and if the mom is exposed to bisphenols excessively during pregnancy, the presence of bisphenols around the cells of the male fetus will affect not the genome of the male fetus, meaning that the XY in every somatic cell will stay XY. The sex chromosomal structure in all the somatic cells will not change, but it is the effect of the male hormones that will not be shown adequately on the male because of the presence of those estrogen mimics that stimulate estrogen receptors and result in feminization of the male. Furthermore, a fourth example and very important example is silencing of DNA. Silencing of DNA, we have already studied silencing of DNA when we were talking about genomic imprinting. And the best way to silence a certain gene is DNA methylation, because when you add a methyl group to DNA, you repress gene transcription. You prevent the transcription of that gene into messenger RNA, and therefore you interrupt the sequence early enough. And then another example of epigenetics is exactly the opposite of silencing of DNA, and it is activation of DNA, because silencing of DNA is one way of inactivation of DNA, but activation of DNA is through the addition of a histone. So we call it histone, through histone, that weakens the chemical attraction between. I'll pause for a minute. Hello again. I'm sorry, I apologize. In number 5, activation of DNA is through methylation of histone. Methylation of histone, as you remember, DNA is wound around or is coiled around histones and non-histones. So here you have the histones and you have DNA that is coiled around. So if this is the histone and this is a piece of DNA that is wrapped around the histone molecule, and in order for this to occur, there should be an attraction between the negative charge on the phosphate group of DNA and the positive charge on the certain amino acids that form the histone proteins. When you methylate the histone, when you add a methyl group to certain amino acids in the histone protein, you didn't make any change in the DNA structure, you didn't make any change in the nucleotide sequence, for example, but all what you did is that you weakened the attraction force between DNA and histones. DNA has a negative charge on the phosphate, histones have certain positively charged amino acids, and there is an electrostatic attraction force between the two. If you methylate histones at certain amino acids, then this methylation will be a barrier between DNA and histones and will weaken the attraction. And if the attraction is weakened, this attraction force is what fixes the DNA in the coiled and condensed form. So if you weaken that attraction, then you're weakening the attraction between DNA and histone. So there is unwrapping of the histone, of DNA, uncoiling of DNA, and this occurs whenever we want to transcribe DNA, whenever we want to uncoil DNA, because when we uncoil DNA, those areas, those uncoiled areas of DNA will be visible for the enzymes of transcription, and this will allow transcription of that segment of DNA into messenger RNA. The best or the most important example for transcription of DNA into messenger RNA is RNA polymerase. So if I ever ask you, why does histone methylation activate DNA? Because it weakens the force of attraction between DNA and histones, results in uncoiling or unwrapping of the DNA. So there is exposure of the nitrogenous bases, and the enzymes of transcription like RNA polymerase will be able to bind with DNA and start transcription of DNA into messenger RNA. Now we're going to talk about something very interesting, which is penetrance. Penetrance means that you might have, we might have 100 people, for example, who have the gene for breast cancer, but only 50% of them show the disease. So in such a case, we say that the penetrance is 50%. Why, if they all have the gene that predisposes them to the occurrence of breast cancer? It all depends upon mainly the environment, the ones who are exposed to environmental factors that are predisposing to cancer, like pollution, for example, or exposure to toxic food or medication or whatever toxins, or increased oxidation for a reason or another, will show the disease. And there might be other genes in a dominant form or in a recessive form that might interact with the gene for breast cancer and either enhance its expression into proteins that cause cancer or diminish and inhibit the occurrence of cancer. Karyotyping is a test that you all know, and karyotyping is a test used to examine and see and visualize the chromosome in samples, in stained samples of cells, and these stained samples of cells can be taken from blood, or can be taken from the bone marrow itself, or can be taken from the amniotic fluid that is present around the fetus in the amniotic sac. The importance of karyotyping is that here, for example, you have a human karyotype that shows you pair number 1, pair number 2, pair number 3, etc., until we reach the last pair, pair number 46, which is the pair of sex chromosomes. And here it's obvious that we have a large X chromosome and a small Y chromosome. So you can see the shape of the chromosomes, you can see the size of the chromosomes, you can see the arrangement of the chromosomes, and you can also see that, of course, we do not do a karyotype during interphase, for example. We usually do a karyotype during prophase. You can see chromosomal mutations. And here we can see an abnormality, but unfortunately, the diagram is not very clear, because if this is pair 18, then this is pair 90, pair 20, pair 21. In pair 21, you are supposed to have 2 chromosomes only. Here you have 3 chromosomes. So this condition is, number 1, considered a chromosomal mutation. This condition is called a chromosomal, not a gene mutation. It is a chromosomal mutation because you have a whole chromosome that is present in addition to the originally present 2 chromosomes. So the total chromosomal number in such a case would not be 46, but it would be 47. So this is number 1. It is a chromosomal mutation in which you have an extra chromosome. And number 2, this chromosomal mutation affects the autosomes, not the sex chromosomes. So we say that it is an autosomal mutation. Number 3, it occurs in a specific pair, which is pair 21, and we have a condition that is called trisomy, because instead of having a pair or 2 chromosomes at pair 21, we have 3 chromosomes. So it is called trisomy. Trisomy is the condition where that is called Down's syndrome or Mongolism, because of the facial features of the individuals that look much alike, whatever or whoever their parents are. And you can have a male Down syndrome or a female Down syndrome, because trisomy affects autosomes, whereas the sex chromosomes might be, like in this condition, XY, or they might be XX. And now we talk about the mutations. As we said, there is a difference between epigenetic change and mutation. In a mutation, there is a change in the genome itself, whether you have a change that occurs restricted to a gene, or a change that involves a whole chromosome, or a change that involves the chromosomal number, but it has to be a change in the genome itself. For example, in the sequence of the nitrogenous bases on the nucleotides, or in a part of the chromosome, or in the number of chromosomes, the number of sets of chromosomes, for example, in the nucleus. Of course, there are factors that help mutation, and we call those factors mutagens. Mutagens are multiple and they are increasing because of the industrial progress. They might be chemicals, they might be cosmic rays, X-rays, ultraviolet radiation. So there are a multiplicity of substances that are called mutagens. And we can classify the mutations in two ways. We can classify them according to whether they occur in somatic cells or in during the formation of gametes. If they occur in somatic cells, then they are not transferable. So the somatic mutations are not transferable. They are not transferable to offsprings because they occur in a somatic cell, not in a gamete, and they will result in the formation of, they will result only in the formation of cancer in the, for example, if there is a mutation here in the skin, then, for example, one might have a cancerous mole, a skin mole, a big a mole that grows rapidly where hairs are present and so on. But if the mutation occurs in a gamete during the process of gametogenesis, then it will be transferable to the offsprings. This is the first way we can classify mutations. The second way we can classify mutations is according to the extent of the mutation. For example, if you have a chromosome and you know that a gene is a part of the chromosome, so if that part is for allele A of the gene, that part is for another allele of another gene, B, C, and D, then if there is a chemical change in that gene only, or in the allele of gene D only, then this will result in a gene mutation. Of course, as you know, when the gene is mutated, then you will have a different protein, and this protein might be a pigment or a hormone or an enzyme. It might be an enzyme, it might be a hormone, it might be a pigment, in which cases it would affect, of course, the phenotype of the individual. Of course, we cannot see gene mutations under the microscope. You need very specific and advanced techniques to decipher the DNA and to know the sequence of the nitrogenous bases, and in order to do a process that is called sequencing of a gene that we will be studying in DNA, the process of sequencing, gene sequencing. But there is another kind of mutation that is more extensive because it might involve the number of chromosomes. You might have one chromosome more, you might have one chromosome less, you might have, for example, duplication of the number of chromosomes, meaning that instead of having one set, you might have two sets, and so on. But again, if the chromosomal mutation is in one part of the chromosome, meaning that it's not a mutation in the chromosomal number, but in the chromosomal structure, then definitely you wouldn't be capable of seeing it under the microscope. So the only ones that can you see under a microscope, like in karyotyping, are the chromosomal number mutations. And now here we have some examples of chromosomal mutations that result in chromosomal aberrations. And when we say aberration, we mean deviation from the normal. Actually, we have 4 examples here. Only 3 are written, so I'll be adding the 4th one, which is duplication. But actually, it is duplication of a certain part of the chromosome. So again, you cannot see it with an ordinary microscope. The first part, let's say or assume that A, B, C, D, E, F is the normal chromosome, meaning that those are genes present on the chromosome, A, B, C, D, E, F. Now, in duplication, it's not duplication of the whole chromosome. Yani if the chromosome were duplicated, you would see another chromosome next to it. But here, what is actually duplicated is the allele. So A has been duplicated twice, okay? And then the rest is okay. We have B as it is, C, D, E, F are not duplicated. So there is a duplication of one gene on the chromosome. In deletion, there is removal of one allele. Here, for example, we have A, B, C, D, E, but F is not figuring, okay? And sometimes the deleted allele might be in the middle, not necessarily at the end of the chromosome. Inversion means that A, B, C have the normal sequence as in the normal gene above, but here, imagine you rotate this 180 degrees. You get the F here and the D here, meaning that you rotate it 100. If you look at that marker, for example, this is the cover, then you rotate it like this, 180 degrees. So what was present to the left would be present to the right, okay? So here we have F now that comes first, and then D comes last. In translocation, you take part of the chromosome, like the first part ABC, and you add it to a totally different chromosome, and this we call translocation. So here we have, duplication is written. Okay, so duplication is a type of mutation that involves production of one copy or more copies of a gene or more than one gene, a region on the chromosome, but not the whole chromosome. Of course, chromosomal duplications or duplication of genes on a certain chromosome will result in the organism having more copies of a certain allele, more copies of a certain allele, okay? And this will result in a more pronounced trait, as we said before, because if you have more copies of the allele, and every allele will be expressed into a protein, then you will have a more pronounced trait because of the formation of more enzymes, hormones, and pigments inside the cell. And this finally results, this will be inheritable, of course, and transmitted to offsprings, and will gradually result in the development of new species or in the occurrence of evolution, as we will see in the unit on evolution. Another last type of chromosomal aberrations is polyploidy. And polyploidy here means that you have the whole set of chromosomes that is duplicated. It totally differs from duplication. In duplication, you take one gene and you duplicate it, or more than one gene and they are duplicated. You find them duplicated. But in polyploidy, it's a whole set of chromosomes that is duplicated. For example, if we imagine that the original chromosomal number of the organism is the haploid number is 3, meaning that the organism in its diploid form has 3 pairs of chromosomes, the pink pair, the purple pair, and the yellow pair. Then you might have an extra set. Yani instead of, if we call this pair, for example, if we call it pair A, and the second pair, pair B, and the third pair, pair C, then instead of having 2 homologous chromosomes as usual in the first pair, we would have 3 chromosomes, you see, in the second and in the third pair as well. So you can express the chromosomal number as 3n. Instead of having 2 sets, you have 3 sets. And if there is a further set, then it would be the chromosomal number would be 4n. So we call this polyploidy, and it occurs in fruits. It's very famous and common in fruits, and it results in having bigger fruits than normal, as you can see on the market, bigger bananas, bigger strawberries, whatever kind of fruit that has a larger size. The sixth type of chromosomal aberration is non-disjunction. I'll first pause for a minute and then resume. Okay, so chromosomal aberrations also include what we call non-disjunction. And we have three examples for non-disjunction of the chromosomes. And all the three examples occur in anaphase, whether anaphase only, just anaphase, which means anaphase of mitosis, or anaphase 1, or anaphase 2. Anaphase 1 means anaphase of meiosis 1, and anaphase 2 means anaphase of meiosis 2. So here we have, for example, in that drawing, we have metaphase. And as the chromosomes are arranged single file, then this means that we are in metaphase of mitosis, and this is indicated by the fact that we didn't write any number afterwards. See here what happened. This is the abnormal chromosome that will not, whose sister chromatids will not separate adequately. So here we found that those two sister chromatids were moved to the poles of the cell. The green ones were also separated and moved to the pole of the cell, as you know, because the cohesion complex in between has disintegrated, but here there was, so this is proper disjunction, but here we have non-disjunction, which means that there wasn't any kind of disjunction between the two sister chromatids of that chromosome, and they all went to one side. And of course, this will result in problems later on, because one gamete will contain only 2 chromosomes, whereas the other gamete will contain 3 chromosomes, one of them being duplicated. But the normal was that every gamete should have received 3 sister chromatids at the end of telophase 1 that we were going to consider whole chromosomes. Okay, the same can occur whenever there is disjunction in case of meiosis. So here, for example, we have metaphase 1 as indicated by the duplicated chromosomes being arranged double file around the equator. What happened is that those two chromosomes did not disjoin. There was non-disjunction, and they both went this side. So again, we have only, instead of having 4 duplicated chromosomes on each side, we have 3 duplicated chromosomes here, and there we have 5 duplicated chromosomes. So at the end of anaphase 1, we have half the gametes having 1 chromosome less, 1 chromosome less, and half the gametes having 1 chromosome more. And then each of these cells will enter metaphase 2. So this is the cell with the 5 chromosomes that enters metaphase 2, and that is the cell with 3. And then disjunction will occur later on normally. So we suppose that non-disjunction occurred once, and that the transfer from metaphase 2 to telophase 2 occurred normally. So you will have half the gametes having n+1 and half the gametes having n-1. N was originally 4. So we have half the gametes that have 5 and half the gametes that have 3. Chromosomal aberrations because of non-disjunction, can also occur during gamete formation, during female gamete formation. As you know, the sex chromosomal structure of a female is XX. And during gamete formation, we are supposed to have one X going to one ovum and the other X going to the other ovum. But what happened here is one kind of sex chromosomal aberration, where the two X's went to one gamete, whereas the other gamete has 0 X chromosomes. And here we suppose that we have a normal male with a normal sex chromosomal structure who is forming the gametes or the sperms during meiosis. So one of the sperms, or 50% actually of the sperms, will contain 50% of the sperms will contain XX chromosomes, whereas 50% of the sperms will contain X chromosome, and the other 50% will contain Y chromosome. So if you do the Punnett square, you will have cases for different possibilities that are all abnormal. And of course, this is expected because we are studying the fertilization of two abnormal ova with two normal sperms. So if XX egg, which is an abnormal egg, is fertilized by an X sperm, which is a normal sperm, we get a female that is XXX or triple X female. And this is an abnormal female because she has an extra X chromosome. On the other hand, if you have this gamete that contains no X chromosome be fertilized by that X sperm, you would have X0 female, meaning that she has as usual 44 autosomes plus XO sex chromosomal structure, and we call this Turner syndrome, and it is a female, but an infertile female with a different body stature because she has one X chromosome less. The fertilization of that 0X ovum with that Y sperm gives the sex chromosomal structure OY, meaning that the whole chromosomal structure would be 44 autosomes plus 0Y. We have said before in part 5 that the X chromosome is important for the formation of certain internal organs. So if an organism does not have X chromosome in particular at all, then this organism would not be viable and would die. On the other hand, if that egg that contains the two X chromosomes is fertilized by a sperm that contains a Y chromosome, then the resultant would be XXY. We would also have a triple sex chromosome case, but in such a case, it's not XXX, but XXY. And since we said before that Y is predominant in male sex determination, so any organism that has Y will be a male, even if it has two X's or three X's. But of course, it will be an abnormal male. It would be an infertile male or infertile male. Why? Because he has an extra X chromosome with extra feminine genes. So non-disjunction is an error that sometimes occurs during meiosis 1. By the way, it can occur, this is not completely accurate because as we saw, it might occur in metaphase, or actually it occurs essentially during anaphase. It occurs essentially during anaphase, so it can occur during anaphase of mitosis or anaphase 1 or anaphase 2, but it's very common during meiosis 1 where the homologous chromosomes fail to separate, as we saw on the diagram. So one gamete will receive the two homologous chromosomes together, whereas the other gamete will not receive any any of the two chromosomes. But when we talk about non-disjunction, we are focusing on one pair of homologous chromosomes. So if non-disjunction occurs in one pair and the two chromosomes go together to one gamete, this does not mean that the rest of the chromosomes have shown non-disjunction. The distribution of the rest of the chromosomes might be completely normal. Here we can see another example that is a bit more complicated of non-disjunction, and that is, as you see here, this shows you that, of course, we are talking about non-disjunction in meiosis, not in mitosis, because you have a homologous pair of chromosomes. And here we have the non-disjunction occurring in meiosis 1, but as I told you, it can also occur in meiosis 2. So this diagram shows you first division, first meiotic division, this non-disjunction, as well as second meiotic division, non-disjunction. In such a case, the two homologous chromosomes went to one of the two gametes, whereas the other one has 0 chromosomes, okay? So during meiosis 2, if we assume that after non-disjunction occurred in meiosis 1, disjunction occurred normally in meiosis 2, then in such a case, what will happen is that these, every one of these two homologous chromosomes would go to one gamete. And instead of having one chromatid, as it should be the case in the 4 daughter cells, and if meiosis 1 and meiosis 2 went normally, we were expecting 4 gametes, and in every gamete we should have one chromatid. Okay, so we would have a non-duplicated chromosome, n equals 1, a haploid, whereas here n should equal 2. But what happened here is that we have 2 empty gametes and 2 gametes, each of them having one extra chromatid. One extra chromatid. Why? Because in meiosis 2, during anaphase, we will separate the 2 homologous chromosomes, and each of them will go to one of the daughter cells. And now if we have a normal haploid gamete that tries to fertilize, and if we have one normal gamete for each of the 4 abnormal gametes, because actually here the 4 gametes are abnormal, these 2 have one extra chromosome, and those 2 have one chromosome less. So here we would have 1 plus 2, 3, 1 plus 2, 3, 1 plus 0, 1, and 1 plus 0, 1. All these are abnormal because these are results of fertilization. They should all be 2n. So here we have 3n, which is trisomy, 3n, which is trisomy, 1n and 1n, which are monosomy. Now here, if in meiosis 1, disjunction occurred normally, but the non-disjunction occurred here. So here we obtain 2 normal daughter cells, and then here this one has or shows normal disjunction, whereas here the 2 chromatids separate during anaphase, but they go to the same gamete. And this is exactly what happened, because you might be wondering why they are not related through the centromere. They separate as if they were going to separate into 2 different daughter cells, and then non-disjunction occurs and they go to one gamete, but now they are no more joined through their centromeres. And again, these are the results of fertilization. 1 plus 1 gives 2, 1 plus 1 gives 2, 1 plus 2 gives 3, and 1 plus 0 gives 1. So because these 2 gametes are normal, so they give normal offsprings, which are disomic, whereas these are abnormal gametes, so they give abnormal offspring, one being trisomic and the other being monosomic. So aneuploidy is any condition in which there is an abnormal number of chromosomes. The abnormal number of chromosomes may be one chromosome more, for example, as in trisomy or in Mongolism, or it might be more than one chromosome more, like in cancerous cells, for example, or it might be polyploidy. Polyploidy means that we have not one extra chromosome, but one set of chromosomes in excess, meaning that instead of having 2n, we might have 3n or 4n or 6n. So there is duplication of the whole chromosomal number. Of course, polyploidy is acceptable in plants, and the plant gives pollen grains and ovules, and it gives fruits that are larger than normal, flowers that are larger than normal. But in human beings, aneuploidy results in abortion. And Hugo de Vries is the scientist that coined the term mutation, not the term polyploidy. And now we're going to study examples of diseases that result from gene mutation. So the diseases that result from gene mutations, not from chromosomal mutations, might be recessive or dominant. So the first one is phenylketonuria, and it is an autosomal recessive mutation, meaning that the mutant gene is recessive and it is present on an autosome, not on a sex chromosome. So phenylketonuria is autosomal and is recessive. And it is due when you have this recessive mutant gene, you cannot metabolize the amino acid phenylalanine. You cannot break down phenylalanine, and therefore there will be accumulation of phenylalanine in the human body, and this will result in mental retardation unless you completely eliminate this amino acid from your diet, which is a difficult task. Cystic fibrosis is a disease of the lungs and of the skin and of all the organs in the body that have ducts in which the human body secretes mucus that is thicker than normal, stickier than normal, and this results in obliteration and blocking of the bronchi, blocking of the pancreatic duct, blocking of the liver, and therefore this would affect the function of all these organs. Again, it is an autosomal recessive disease, and it might be lethal, leading to death of the organism. And it has been shown that cystic fibrosis is the most common lethal genetic disease in the United States, and it is common, or actually the frequency in Caucasians is 1 out of 25, which means 4%, and this is a high percentage. And then we move on to show more examples of autosomal diseases resulting from gene mutations. So we have the Tay-Sachs disease, and it is very common in Ashkenazi Jews. It was written incorrectly on the slide, so please correct it. And I would like you also to copy this sentence on your slides, because it was written very shortly in a way that might lead to understanding. Again, Tay-Sachs disease is an autosomal disease where the mutant gene is recessive and is present on the autosome. It is common in Ashkenazi Jews, as I said before. The symptoms in Tay-Sachs disease are mainly neurological symptoms in the form of seizures, convulsions, blindness, and maybe also early death. The recessive mutant gene results in failure of formation of a very important enzyme, and that enzyme is important to break down a certain type of lipids in the body, and these lipids are called gangliosides. So if these gangliosides are not broken down, they accumulate in the human body and they result in mental retardation. Sickle cell disease is again an autosomal recessive disease, and we will be discussing it in detail in DNA. And as you know, there is dominant gene for normal hemoglobin and a recessive gene for sickle cell hemoglobin. If you are homozygous dominant, then all your hemoglobin is normal. If you are heterozygous, having the dominant allele and the recessive allele, then you have a small fraction of your hemoglobin that is abnormal. You are a heterozygote individual, having a small amount of your hemoglobin that is abnormal. So you show the symptoms only when you are exposed to severe depletion of oxygen, like when you climb a mountain, for example, a very high mountain, or when you exercise heavily. But if you have both recessive mutant genes, you are a homozygous recessive individual, so you are no more a carrier, but you are a diseased person. So you have the sickle cell disease, and not the sickle cell trait. And in such a case, most of your hemoglobin will be abnormal. And this abnormal hemoglobin, when you are exposed to low oxygen tensions, will be converted from globular hemoglobin to elongated hemoglobin, meaning that it will be denatured. And this, of course, will deform the RBCs. So you would have deformed RBCs. They would look like the sickle with which we cut plants, and those deformed red blood cells will stick to each other, forming rouleaux, and those rouleaux can obliterate the blood vessels, resulting in organ failure. You might be wondering why, if in case of the carrier state, why does the recessive allele show its effect, although we have a dominant allele here? And this is a new non-Mendelian case that we haven't mentioned before because it's not mentioned in your curriculum. That is called incomplete dominance, not lack of dominance. Incomplete dominance, so we previously saw lack of dominance, in which there is blending of effects. We saw co-dominance, like MN, both show on the phenotype, or AB, yes, AB. And then we have incomplete dominance, where the dominant allele masks the recessive allele, but the recessive allele sometimes shows partly under certain environmental conditions, which are like, for example, the low oxygen tension. People who are heterozygous for sickle cell trait are resistant to malaria, and we call this the heterozygote advantage, and you will be studying it in evolution. They are carriers. This is something that's not good, but this gives them the privilege to be resistant to malaria. And therefore, in areas that are endemic for malaria, we will find that heterozygote individuals, S capital, S small, for sickle cell anemia have a high percentage in the population. Huntington's disease is an autosomal mutation, but it is a dominant mutation that results in early death because it leads to degeneration of the nerve cells. There are other gene mutations that are sex-linked, okay? So we have hemophilia, we have color blindness, and we studied them before when we were studying sex-linked inheritance, meaning that the recessive mutant gene will be on the X chromosome. And in hemophilia, of course, if a female, for example, has both recessive mutant genes for hemophilia, if it is hemophilia A, then she will have a defective factor 8. If it is hemophilia B, she will have a defective factor 9, and both factor 8 and factor 9 are important for blood clotting. Duchenne dystrophy is another sex-linked mutation, gene mutation, but it results in progressive weakness of the muscles that might lead ultimately to death if it affects the muscles of breathing, because the patient would be unable to breathe. We have just seen chromosomal mutation. We studied Down syndrome, Turner syndrome, and Klinefelter's syndrome. And we will stop here because this is the last item. We will be discussing it later on in part 6 or in the classroom. And this is the last thing we will be discussing in heredity. Hope this video was useful, and see you, inshallah, in the next presentation or in the classroom on Thursday. Bye.